Why restriction endonucleases are known or named as restriction endonucleases, that we have to know first. Restriction because they are part of a particular system of prokaryotes. That system is known as Restriction-Modification system, or in short, RM system. So what is RM system? The restriction system, actually in 1950s, scientists noticed that the bacteria have the ability to restrict the infection of bacteriophages. You know that bacteriophages are viruses. Those viruses can attack a bacterial cell and can kill the bacterial cells. Okay, so in the bacteria, like E. coli, Escherichia coli, have the ability to restrict or inhibit the infection process of some bacteriophages. The mechanism was unknown, but that observation was there in 1950s. Okay, so later, two scientists, Arbor and de Siox, in 1962, actually discovered or clarified the molecular basis of that phenomena by observing that that bacterial cells have a group of enzymes. That enzymes can digest the viral DNA. Here you can see in this figure that viruses are inserting their DNA within the bacterial cell. Here the DNA is inserted. So after insertion of the DNA are recognized by restriction endonucleases in different locations, and the DNA is fragmented. So that is why the viral DNA is unable to replicate or proliferate into new viral particles. Okay, in this figure also you can see that this virus is inserting its DNA, but the DNA is immediately recognized by the restriction endonuclease, this yellow colored one. Okay, so this is digesting the DNA, so the virus is unable to infect the bacterial cell. Clear? But here another problem was noticed by the scientists, that if the enzyme is able to digest DNA, so how the bacterium is protecting its own DNA? It is this particular, in this figure, it is shown the chromosomal DNA of the bacteria itself. So restriction endonucleases can cut the bacterial own DNA also, but that is not happening. So there must be some other mechanism by which the bacteria is protecting its own DNA, and that system is known as modification system. Okay, modification system is nothing but having another enzyme that is known as methylase. Methylase. Okay, so what methylase does? Methylase adds a methyl group, CH3, methyl group to nucleotides in the DNA chain. So here in this figure you can see Me means methyl group. So methyl groups are added into the bacterial own DNA. That is why the restriction endonucleases are unable to recognize this methylated DNA of the bacteria. That is why they are unable to digest their own DNA. Okay, so that is why these two systems, that is restriction means the endonuclease function, and the modification means this methylase function. These two functions are integrated to each other to generate this restriction modification system.